Hey folks, we're going to take a look at glycolysis, decarboxylation of pyruvate, Krebs cycle, and the electron transport chain. Starting out with a little bit of orgo on glucose. Here is alpha glucose, and this is beta glucose. They exist in equilibrium with the linear form, but they predominate as far as concentration goes. These are called anomers. And the way this works is, although people use up and down for the hydroxyl on the anomeric carbon, which is the carbon that has two single bonds to oxygen, the appropriate way of doing it is looking at cis or trans. So this is the anomeric carbon, the carbon with two oxygens, one here and one here. This is the anomeric carbon, number one in this case, but it could be number two. And this is the anomeric carbon. It derives from the formation of the hemiacetal from this carbonyl right over here, which would also be carbon 1. The beta anomer has a relationship which is cis to the terminal carbon. In this case, the terminal carbon will be carbon 6. So this is 1, 2, 3, 4, 5, and 6. And what we have here, this is trans, that's the alpha anomer, and the beta anomer is going to be the cis relationship. And I know what you're saying. You say, I always do it up or down. This is down, this is up, and I get it right every time. But you won't always get it right every time. So if you're not sure, this is the right way to do it. Okay, and the other thing we need to say is that the concept of an anomer is often confused with that of an epimer. So we're not going to talk about epimers right yet, but remember that an anomer is going to be an absolute configuration on the carbon with two oxygens, and an epimer is going to be something else. You may have also noticed in the chair form, this molecule of glucose has all these groups that are going to be equatorial, which means it's going to be super stable. And that's an interesting characteristic of glucose. Up over here, this process of equilibrium, where the equilibrium changes, this is called mutarotation. And the idea of mutarotation is if you change the conditions, the equilibrium will change. And you've got a carbon here, which is going to be chiral. Here it's achiral, and here it's chiral, the opposite configuration. So if you change conditions, its optical activity will change. And that process of being able to view the change in optical activity is called mutarotation. It's done with a big old polarimeter tube. Okay, as far as glucose goes, it can be present as a polymer in starch. And you have 1,4 linkages here, 1,4. And in this case, because this relationship over here is going to be trans, it's an alpha linkage, 1,4 alpha. And in the case of cellulose, which is the same except for this linkage, we're going to have 1,4 of beta. So that's the rough idea. Now let's take a look at something a little scary. So what I'm going to do is I'm going to take a look at metabolism. Now, the parts of metabolism which are on the MCAT are everything in white, but not everything, all the details, but the general idea is what's on the MCAT. Right over here, we have glycolysis. And up over here, we have glycogen, which is a storage form of glucose, which you can turn into glucose. And then you can turn glucose through glycolysis into pyruvate. Pyruvate can be turned into lactate in something called fermentation, or it can go into the Krebs cycle as a result of a decarboxylation. And when it decarboxylates, it forms acetyl coenzyme A. And the CoA comes from fatty acids. That's this over here. It goes into the Krebs cycle, and the Krebs cycle produces molecules that have high energy that make ATP. And that's called the electron transport chain, also known as oxidative phosphorylation process. It's not shown here because it's not a chemical reaction. It's a different form of reaction. These are just chemical reactions here. Now, just for your information, hexose monophosphate shunt, that's the same thing as the pentose phosphate pathway. It's just another name for it. That's where you manipulate sugars to make a whole variety of sugars. This over here, not on the MCAT. Fatty acids on the MCAT. Urea cycle, anything involving nitrogen on the MCAT. So here's just a rough idea of where everything is. As far as glycogen goes, what happens with glycogen is it's a polymer of glucose, but it involves bonding of the 1,4 and also the 1,6 variety. You're not actually responsible for its structure, but you are responsible for its breakdown, in which if you don't know its structure, you can't figure out how it's broken down. So that's for another time. But that glycogen is right over here on the top, leads into glycolysis. So now it's time for glycolysis. This is occurring in the cytosol. We start with one molecule of glucose, that's 6 carbons, and we use 2 ATPs. And what we do with those ATPs is we phosphorylate these sugars right over here, and that gets the molecule ready for further reactions. It provides the energy. We then have this molecule with 2 phosphates, 6 carbons. It breaks into 2. And what happens is one of those 2 is in equilibrium with the other, and it's going to go straight down to pyruvate, which is going to be a 3-carbon sugar. So we'll have 2 3-carbon sugars coming from the 6 carbons of glucose. The first thing we're going to do is we're going to make NADH. So per glucose, what we're going to do is we're going to make 2 NADH from NAD+. And we're also going to do a phosphorylation right over here. NADH is a high energy reduced molecule. When you've got hydrogen on the right-hand side, that's a reduced molecule, usually. And what happens is that will be used in the electron transport chain to make ATP. We're also going to make over here 4 ATP. So we're going to put 2 in and get 4 out. So that's going to be a net of 2 ATP. Now, when we say substrate level phosphorylation, that means that the phosphorylation, the addition of phosphate, was done chemically using bond energies. That's what substrate level phosphorylation is. And isomerization means there's been no change in the molecular formula, just the molecular structure. Dehydration, loss of water. And then, of course, the second substrate level phosphorylation. Now, you don't need to know all the molecules here. You don't even need to know all their names, but you do need to understand that what we've accomplished is we've made 2 net ATPs and 2 NADHs per glucose. Now, all of this has occurred in the cytosol. And what happens is the pyruvate that we made, the 2 pyruvates, go straight into the matrix of the mitochondria for free. But what will happen is, I forgot to turn this off. What will happen is that the NADH is not able to get into the mitochondria. So what ends up happening is there's a process called the glucose phosphate shuttle. And you don't need to know this. All you need to know is that every NADH that's produced in the cytosol produces an FADH2 inside the mitochondria. And this involves energy loss because NADH has more energy than FADH2. So this is a process that you don't need, but you do need to understand that what happens is for every NADH, we get an FADH2 in the mitochondria. So for glucose, we make 2 NADH, we end up via the shuttle with 2 mitochondrial FADH2s. So that's kind of the bookkeeping behind this. All of this has been in the cytosol, and we're now going to move into the mitochondria right over here. So what happens is that I went the wrong way. Okay, so what happens is we have our molecule of pyruvate. That's a 3-carbon molecule. And you actually don't need to know its structure. What will happen with this is we're going to take that pyruvate and we're going to generate more NADH. But here's the deal. Everything that I did over here is going to be per glucose. And over here, everything I'm going to do is per pyruvate. So per glucose, all of this is going to be ×2. Now, that's not so hard to deal with, especially because sometimes they ask about per pyruvate, sometimes they ask about per glucose, so it's easy to work out. So what will happen is we make 1 NADH for this pyruvate, but actually 2 NADH per glucose. And we also have 2 carbon dioxides leave per glucose. That's 1 CO2 per pyruvate. And what happens is the 3-carbon pyruvate becomes a 2-carbon acyl group. So, I mean, if you're into orgo, the acyl group that we're talking about occurs very commonly in biochemistry. It looks like this. So that's the acyl group we're talking about. And what happens with this is it's going to go into the Kreb cycle, also known as the citric acid cycle. Oxaloacetate is this molecule here. We'll see it. It's probably a good idea to know it. When we add the 2 carbons here, we get citrate, which is the name of the cycle. You don't need all of these, but you need to be familiar with their names, which we'll talk about. Bottom line, though, as far as adding this up, we're going to go ahead and produce 3 NADHs per pyruvate. We're going to produce 1 FADH2 per pyruvate, and we're going to produce 1 GTP per pyruvate, which becomes an ATP. So the bottom line here is we have all these molecules, and these molecules are going to be used to make ATP. GTP does it pretty much directly, but the rest of these use something called oxidative phosphorylation. And the principle here is these high energy molecules take their electrons and they're going to use them to pump hydrogen ions outside the matrix. When the hydrogen ions go back into the matrix, they're going to allow the generation of ATP through a molecule called ATP synthase. And the bottom line is that for 1 NADH, we can produce 3 ATP. For 1 FADH2, we can produce 2 ATP. So NADH is more energetic. And this process occurs as a result of taking electrons and moving them down the line, pumping hydrogen out. And also in the last step, the final acceptor of these high energy electrons is going to be oxygen. That's why we need to breathe in oxygen to make this process possible. And what will happen is we're going to generate water. So when you think about it, we breathe in oxygen and we breathe out water, vapor, and carbon dioxide. Why? Because that's all about metabolism. That's what goes in and that's what goes out when we have anaerobic respiration, aerobic respiration, meaning oxygen is involved. Okay, so the thing we're going to do is we're going to take a look at all this information here and add it up, because that's one of the things that they like to do. So there's our glycolysis, and then over there on the left, that's going to be our Kreb cycle and the electron transport chain. So here's what will happen. We have per glucose a net of 2 ATP. We lose 2 ATP here, that's right here, and we're going to gain 4 ATP total. So that's a net of 2. And that takes care of Krebs. I'm sorry, that takes care of glycolysis. As far as the Krebs cycle goes, per glucose, we're going to end up making 2 GTP, which are equivalent in energy to ATP. Now, if we look at oxidative phosphorylation, that's everything occurring here in the electron transport chain. So what will happen is we're going to make 4 ATP from the 2 NADH in glycolysis. That's these 2 over here. But you say, hey, you're not supposed to make 4, you're supposed to make 6. But you only make 4 because you don't have NADH, you have FADH2. So you end up having 4 FADH2, and that's going to give you, let's see if I did that right. 4 from the 2, yeah, you're going to have 2 NH2 right here, and 2 times 2 is 4. Okay, sorry about that. Hope that made sense. Over here, we're going to get 6 ATP from the 2 NADH and the decarboxylation of pyruvate. So this is times 2. So what will happen is this will go ahead and produce 2 NADH, and then 2 NADH will produce 6 ATP. And then we're going to have 4 ATP from the 2 NADH2 in the Krebs. So here's the NADH2, 1 per pyruvate, 2 per glucose. If you have 2, that'll make 4 ATP. And finally, the most ATP we're going to get is going to be right over here. What's going to happen is we're going to get 18 ATP from the 6 NADH in the Krebs cycle. So we have 3 NADH per 1 pyruvate, that's 6 NADH per glucose, and then that's going to be 6 times 3, or 18 total. And that gives us 36 net ATPs per glucose for this process. So on the MCAT, they ask questions that are like this. Sometimes they ask you to start in a particular place, either over here or over here. And we'll look at all this in more detail. Even though you don't have to memorize it, they present you with the information. Being familiar with the territory will make it much easier. Okay, so let me go ahead and get rid of this right now. And what we'll do is we'll take a look at glycolysis. So the only thing I want to point out here is that there is a parallel process to glycolysis, which is called gluconeogenesis. And in this case, we're actually making glucose as opposed to breaking it down. And it's almost identical. The only real difference is some of the enzymes are different, most are the same. And oxaloacetate is a step in gluconeogenesis and is a molecule present in the Krebs cycle, which you should know about. It's the starting point, really. But this pretty much is the layout. You don't need to know all the names. You don't need to know all the structures. But I just wanted to show you how it looks. Okay. Now let's go over here. Okay. So this is the Krebs cycle here. And on the MCAT, they have frequently presented the Krebs cycle and asked questions. How many carbons will a molecule have? Is it an oxidation? Is it an isomerization? What's going on? And you don't need any information to do it because they give it to you. But I just wanted to show you what the idea here is. Essentially, you should know that oxaloacetate is the starting point where we're going to go ahead and add the acyl group in acetyl coenzyme A. Just the acyl group, which you see right over here, is going to enter the cycle. When it enters the cycle, we get citrate. That's the first molecule here. And I wanted to show you some of the things that they will typically ask about this. And by the way, they're not going to make any modifications to the cycle. It's going to be this. They don't do that. It's going to be just what we have. So a little familiarity will help. This is called dehydration. You're going to be losing water. And sometimes they ask something like, they show you it's a dehydration by showing a water being leaving, and then they ask you what the molecular formula of this is. So all you do is you get the molecular formula of citrate, and you basically subtract H2O. And it's that easy a question. But the other thing they'll do is they'll say, well, from citrate over here, we lose water. And over here, what's going to happen, we're going to gain water. And then they'll ask you, well, what's this process going from here to here? And so you can safely say that process must be isomerization, because you didn't gain anything, you didn't lose anything. That must be isomerization. And you know it's not going to be the same molecule, because that would defeat the whole purpose of the cycle. The other thing that occurs quite a lot here in these two steps is we're going to have oxidative decarboxylation. So the thing is that when you have oxidative decarboxylation, you're going to have a loss of the carbon dioxide. But along with the loss of a carbon dioxide, you're going to add a bond to an electronegative atom, such as an oxygen or such as a sulfur. So what happens is we're going to be losing a CO2 right over here. So if we look at where we're losing our CO2 over here, let's see, I think we're losing the CO2 like this. It's gone, you see? So that's our decarboxylation. But then it's oxidative because there's one bond to O here, that's electronegative, and here there's two bonds. So when you add a bond to O or S, something electronegative, they're called heteroatoms, more electronegative than carbon. What will happen is you're going to have an oxidation. Now, in this process, we also see well, you go from NADH2 over here, and you're going to go from NADH2 and NADH over here. So these are molecules that are going to be reduced. They're going to be produced. So these are reduced forms. So whenever you see these, if you don't have the structure, you know that the molecule must be oxidized. The molecule must be oxidized if the counterpart, the other half reaction, is going to be reduced. And just to show you over here, what happens is here they're going from a CC double bond to a single bond with an OH. Now, what happens is, let's start over here. A double bond is more oxidized than a single bond. So this is oxidation, and we're not surprised because we see NADH2, that's the reduced form, so this molecule must be oxidized. In fact, this is all about oxidation. We're burning fuel, like burning wood on a fire. And over here, we have a double bond, and then up over here, we have an OH. Now, you may not be familiar with a certain system for determining whether something is oxidized or not. But with a double bond carbon, you can use a 1/2 as a way of figuring out how oxidized it is. I'm not calling it an oxidation number. It's a system. It's a great system. And then over here, what we have is an OH. Now, with the OH, when you have a single bond to an electronegative atom, that's equal to 1. And over here, this would be equal to 0. That's just a hydrogen. And the idea here is the total here is 1, and the total here is 1. And what that means is this is not oxidation or reduction. And just to make it clear, if we had a triple bond, we would have over here a 1 and a 1. So it's a very simple system. I'm going to give you a little bit more to it. If you had this, this would be a 2. And if you had this, this would be a 3. So the rules work like this. If you have a pi bond, it's a 1/2 and a 1/2. If it's a double pi bond, it's 1 and 1. If it's a single bond to an electronegative element, it's a 1. If it's a double bond, it's 2. If it's 3 bonds, over here, it's 3. And you add them up. And if the number goes up, it's oxidation. If the number goes down, it's reduction. And if nothing happens, it's neither. And of course, if you think about it, here, we're really adding water. Water is not an oxidizing or reducing agent. So we would expect this to be neither an oxidation or a reduction. So that's the kind of flavor of doing this. They don't necessarily give you this whole thing. They give you portions of it, ask you about it, but along these lines. So it's really an understanding of chemistry and biochemistry. What they did here was they presented the NADH and the FADH2 as coming off the cycle. So it's the same idea, it's just presented in a different way than the slide we saw before. Okay, so what we're going to take a look at now is how do you control glycolysis and the Krebs cycle. So the idea is, if you want to do glycolysis, you want to make ATP. You want to make ATP. If you don't need ATP, you don't want to do glycolysis. So what happens is that if we're taking glucose and making glucose 6-phosphate, that's the first molecule after glucose, and we have a lot of this, then that's going to downregulate the production of glucose 6-phosphate. In other words, we got too much product. That means that the enzyme hexokinase will be downregulated. We'll say, turn it down. On the other hand, if we have phosphate, phosphate's what we need over here. So if we have a lot of phosphate, we have what we need to make this, then that's going to upregulate it because it means we have the raw materials. But phosphate is a simple molecule. It's not like glucose 6-phosphate. So that's the reason for the difference. Now, if we come over here, what we're looking at is, we're looking at the fact, this is pretty straightforward. When you have a lot of AMP, a lot of ADP, you don't have a lot of ATP. So that means that you would upregulate this. You would say, I want this to go faster because we don't have enough stuff here. Now, if we look here from the Krebs cycle, the Krebs cycle is going to make ATP. And when it makes ATP, it's also going to use citrate. Citrate is the first molecule that's made after we add the acetyl-CoA. So if we have a lot of citrate and if we have a lot of ATP, we're going to downregulate the enzyme phosphofructokinase because we don't want to make more than we need. And then up over here, what happens is, this is along the way in glycolysis. So if we have a lot of fructose 1,6-bisphosphate over here, we're on our way. We've got raw materials that we can use. So we're going to upregulate way over here. We're going to say, okay, let's finish the process. So all this is pretty logical. And over here, if you've got a lot of ATP, you're going to downregulate because you don't need to make more stuff. So that's the basic idea here, along with one more thing that wasn't mentioned, but it is part of it. Every time we are going to make an ATP like this right over here, or these are the 4 ATP we make, if we have a lot of ATP, that step will be downregulated because we don't need more ATP. That isn't mentioned because these are not considered major control steps, but they are part of the deal. Okay. Now, on the bottom over here, what we have is we have what's called fermentation. And I'm going to talk about more of that in another video, but it's a pretty simple process. If NAD+ is required for glycolysis, and glycolysis is pretty much required for electron transport chain, then if you run out of NAD+, you're in bad shape. So what happens is fermentation takes pyruvate, turns it into lactate, and produces NAD+, which can then be used to continue the process of glycolysis. Now we're going to look at something similar. Here we have glucose, and here's gluconeogenesis. We're not going to look at that, but it was a nice slide. So fructose 2,6 bisphosphate is a side product of glycolysis. It's not in the direct chain, it just comes right off over here. But what happens is you get a whole lot of it if glycolysis is cooking. So what happens is, if you've got a lot of this, you're going to make this go faster, like this. And over here, if you have AMP, that means you don't have a lot of ATP. So you're going to make this go faster. And if you don't have a lot of ATP, you make this go faster because you want to make ATP. Citrate is the first molecule really that in the Krebs cycle. So if you don't have a lot of citrate, you want to make this go faster so you can make more. And as far as H plus goes, H plus is part of the electron transport chain. And what happens is you're using H plus to make ATP. So if you don't have a lot of H plus, you want to make some so you can get more ATP along the way. So that's why this would go forward. So I know I'm going through it fast, but it should all make sense because it's very logical. Now here, fructose 1,6 bisphosphate, that's this stuff. That's halfway through the process. So what ends up happening is, if you've got a lot of this stuff, you want to keep going because you've already invested in it and you're ready to go. If you don't have a lot of ATP, you want to go and make some more. And alanine is kind of interesting. It turns out that pyruvate is just one step away from alanine. So if you don't have the amino acid alanine, you can make pyruvate, and in one step, you can have alanine. So that's why alanine is going to be over here. And don't worry about all this stuff, gluconeogenesis, although it's kind of the mirror image. Okay. So let's talk about the process of regulating the Krebs cycle. So here we have pyruvate. And what happens is, if you have a lot of ATP, you're going to shut it down or slow it down because that's what the Krebs cycle does. It makes ATP. And then over here, we have acetyl coenzyme A. If you've got a lot of that, you're going to shut the process down. NADH, well, that's what you make in the Krebs cycle. As far as fatty acids are concerned, fatty acids make acetyl coenzyme A. So that's the reason why if you have a lot of these, you'll shut the process down because you don't need any more. On the other hand, if you have AMP, it means you don't have a lot of ATP. So you're going to go ahead and make the process continue. Coenzyme A, well, that's what you need to do the Krebs cycle. NAD+, that's what you need. And calcium, it turns out that when calcium is released, muscular contraction begins, uses up a lot of ATP. So when you have calcium 2+, it'll use up a lot of ATP, so you got to make more. So that makes coenzyme A right over here. And then over here, what happens is, if you've got a lot of NADH, well, that's what you're making, so you don't need more. Succinyl-CoA, that's along the way in the process, so you don't want to make any more. Citrate, that's right over here. Don't want to make any more. ATP, if you got a lot of ATP, you don't want to make any more. So these work pretty much the same way. You can look at these, but there's really no difference. So the concept here is if you've already got what you need, you slow the process down, and if you don't, you speed the process up. Okay. So the final thing I wanted to talk about is the electron transport chain. And I think what I'm going to do is I'll pull up another slide here so I can talk about it here. Okay. Yeah, that's good. Okay, so the idea behind the electron transport chain is you're going to take molecules of NADH and FADH2, and those molecules are going to use their energy by passing electrons to different protein complexes 1, 2, 3, and 4. And they will cause hydrogen ion to go into the mitochondrial matrix. So to come out of the mitochondrial matrix, excuse me. So that's what's occurring here. They use that energy to pump them out. When they pump them out, they go back in through a transmembrane protein called ATP synthase, which generates ATP from ADP and pyrophosphate. And it does so by using hydrogen ion, the high concentration of hydrogen ion that has been pumped out of the matrix. This is called oxidative phosphorylation. It is a non-chemical mechanism. It doesn't involve bond energies. And it produces the most ATP in this entire process. Now, as far as the numbers go, you don't need the numbers, but the way it works is it takes about 4H plus to make an ATP. I was just curious about that. And it varies with the pH and other conditions, but that's the idea. And the molecules that we see here have a certain structure, which we'll look at when we talk about organic chemistry, quinones, plastic quinones, which hold on to and pass energy along. But that's the rough idea on how this works. And I hope that you enjoyed the show. Please come back for more. Thanks a lot.